This new method for analyzing reverse transcription can tell us new things about the field of retrovirology. For example, we can learn about the details of how cellular restriction factors work or how antiretroviral drugs inhibit HIV-1 DNA synthesis.
The main advantage of this technique is that it not only provides information about the sequence of nascent reverse transcripts, but allows mapping of their precise 3' DNA termini at single nucleotide resolution.
The enrichment of HIV-1 complementary DNAs from whole cell DNA of HIV-1 infected cells is done by hybrid capture and should be carried out in a PCR workstation. Prepare a master mix of magnetic streptavidin beads by pipetting 100 microliters of beads per sample into a single microcentrifuge tube. Place the tube on a magnet suitable for microcentrifuge tubes. After the beads have settled toward the magnet side of the tube, remove the storage buffer, remove the tube from the magnet, and resuspend the beads in 500 microliters of bind and wash or BW buffer. Place the tube back on the magnet, wait for the beads to migrate to the magnet, and remove the supernatant. Remove the tube from the magnet, add 500 microliters of casein solution, resuspend the beads, and incubate at room temperature for 10 minutes. After 10 minutes, wash the beads with BW buffer. Place the tube back on the magnet, remove the supernatant, and resuspend the beads in 500 microliters of BW buffer. Add 50 picomoles of each captured biatenylated oligonucleotide per sample. Incubate at room